Hello everyone, just as a reminder, this is going to be part one of two parts for nervous system. And so this is a lot of this is a redo. We're only really adding the last four slides, I believe, and the myelination and action potential, and otherwise that should be the only new material, so hopefully you've been studying and this video for this beginning portion sounds like really easy. Alright, so when you're looking at the human body, right, here's our brain, here's our spinal cord. The brain and spinal cord make up the central nervous system. You have to think about this, that this is who you really are. Despite, what you look like, your face, your hair color, who you really are is really determined within this space. And this is what you really think, your memories, what you process, all the information that comes in. This is what fundamentally decides what you're going to do. And at the end of the day, it even controls the stuff that you have inside of you, your heart, your lungs, digestional system, things that you don't even want to worry about. So everything that you are is really this little object here, this brain. And so now the spinal cord goes down, and you have to think about the spinal cord as an organization of nerves. It organizes the nerves all the way leading down, so that if you want to send a signal from the brain, it goes down through the spinal cord and exit out over here if you want to control something here. Or it exit out over here because you want to control something there. So the spinal cord in your head should be just a freeway passageway that allows nerves to exit in and out. Now, information processing occurs in the brain. Peripheral nervous system refers to all the nerves that are outside of that spinal cord. That's it. The second we leave the spinal cord, peripheral nervous system. Enteric nervous system has to do with digestive tract organs. Do you remember the myenteric plexus? Right? That's the nerves that are in between the smooth muscles, that muscular layer, circular and longitudinal, that allow you to move, to do peristalsis within your small intestines. So those signals come from your brain. And so that's what we consider enteric nervous system when it's going to control things in your digestive system. And next, you have to think about it in two directions. That the brain, you can go into the brain, right? If you touch something on this fingertip, if you're following my mouse right now, that signal goes up, goes into the spinal cord, and then goes to the segment there that's meant for touch. And so you're able to process the temperature of things, how cold something is, because it traveled into your brain. We call that traveling into your brain afferent. Afferent. A with an A. Efferent would be the opposite, going in the opposite direction. So it makes sense now that the word sensory division, you should know what your senses are: smell, taste, sight, balance, hearing, touch. And those are senses, hence part of your sensory division. And that means that everything that you detect, like touch, for example, will go into your brain afferent. If you see things, right, you're seeing this man is seeing this, you see it, it goes into your brain and then goes to this part of your lobe. But it's still going in. Smell things, it goes in, but through your nose and into your brain. Taste things, you taste it, and then it goes into your brain. And so you can see that all of the senses are essentially a part of the sensory division. That means going into your brain. Pretty easy, right? Motor is going to be exactly what it sounds like. Whenever we heard of motor in anatomy, you should automatically jump to muscles, period. Whether it's smooth muscle, whether it's cardiac muscle, whether it's skeletal muscle. Motor means muscles, the three muscles. And you can divide them up because somatic nervous system, this means body, somatic. So body nervous system is the voluntary nervous system, which means all the skeletal muscles. All the skeletal muscles are under your control, and they're part of the somatic nervous system. The other two, smooth muscle, cardiac muscle, these are under the control of autonomic nervous system, which makes it pretty easy, right? These are the ones that you don't do anything, right? We already drilled on this a bunch of times. Smooth muscles, do you control them? No. Cardiac muscles, do you control them? No. Hence, they're going to be part of the autonomic nervous system. Sort of rhymes with automatic. Be careful with that. But autonomic. And then some other things like glands, and then the regulation of our adipose tissues. But that's a little bit further down the line. But otherwise, glands, we'll see a little bit later. Adipose tissue, not so much. And so try to be, before we move on to that. And so now that when you look at this table, I want you to be able to understand it. When you look at receptors, this is going to be everything that's sensing. We're seeing how these two things, these two divisions work together. Sensory division and motor division. Sensory division now, right? You're sensing things, a receptor's touch. It's going to go up, 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 and you'll notice that we're in this yellow segment of this table now. This is the peripheral nervous system, meaning all the extensions, all these yellow cables coming out of this spinal cord here. And so this peripheral nervous system is going to continue to be peripheral nervous system until we hit the brain. The second, and then you see that that's the role of the peripheral nervous system, to send these antennas out, really, right? Like these nerve cells. Extend them out, send the signal in, and finally arrive into the central nervous system where we can go into the brain. And then now we are part of the central nervous system. And then we will be able to process that information. Next, you'll see now that you processed it, right? You detected it out here, you send a signal out, and then it goes into your brain. And now motor division, again, this is going to be the motion to move things. So you're going to send a signal out this way. Let's say that you felt something that's really cold and it's uncomfortable. You don't want to feel it. It goes in through your peripheral nervous system, into the CNS, your spinal cord, then your brain. Then you process, this is really cold and uncomfortable. What do you have to do to move your hand? You have to send a motor command. You have to move a muscle, maybe around here. So from that division, another part of your brain will say, all right, time to move it. Sends it out. It exits this location. And then it now enters in the peripheral nervous system, right? Because we came out of the brain, we came down through the spinal cord, exit through the peripheral nervous system. Then we go to the muscle here, and now we move our muscle. We contract our biceps brachii, right? And now we can stay away from whatever was bothering us. So that's what this is, motor division. You're seeing a signal going into your brain, it's coming out to the brain. And now sometimes, this one's the obvious. This is the one we just did, somatic nervous system. You decided, I wanted to move a muscle, right? Skeletal muscle. Autonomic nervous system. This is a little bit different because there are two things you have to know here. Is that autonomic is things that you do not control, and that things that you do not control are under the control of two different systems. And that's going to be the only hard thing about this. Is that there are some things that you do not control, and that's under parasympathetic and sympathetic. What I made you write in class in sympathetic division is that it's fight or flight. This makes all the organs, smooth muscle, cardiac, the glands, adipose tissue, it makes every organ in your body work together. Fight or flight responses. Okay? Like that nervous feeling you get. It brings it all the body there because it wants to survive whatever it is. And it may not make sense why you need to pee when you're nervous, but if you're like running away from a predator and a little bit of pee's on you, maybe they don't really like that flavor, And so that's a deterrent for a lot of animals. So it's still biologically within us. And now parasympathetic system is going to be the division of things that you don't want to take care of, like the boring stuff. It's not just fight or flight, it's boring stuff for parasympathetic. They take care of your digestional tract movements. They can take care of your heartbeat, your lungs. Like they just take care of things that you don't want to worry about all the time. But you can see that they're controlling parts of you that you don't really have direct access to, right? And that's it for this slide. Learn this slide, and all I really did was sum up all these things with this picture. So whenever if you haven't learned by now, make sure that you're always looking at each picture, and then you are able to explain each picture to somebody. Okay, and don't move on before you can answer these questions. And so we have to address what those cables are, what's in your brain. And what you see in your brain, when that first picture we saw, all those nerves, peripheral nerves, and the spinal cord and the brain, they are all made up of these cells. So you have a cell body, you have a nucleus, you have a nucleolus, just so you're seeing that this is the same stuff that you've seen before in other cells. Some things that are a little bit different is that their rough ER is broken or fragmented into little pieces called Nissl bodies. And so these, instead of it being like it was before, they're sort of fragmented. And they also have free ribosomes. So this is a cell. Now, something that makes it unique from other cells is dendrites. Dendrites are the receiving part of a signal. This is where they receive signals. And then the axon, you can see that there's always going to be a longer extension. You can see it goes on, and it's synapses. Synapses means to connect with something else, and it's the word I'm going to start using. Log it away. So this nerve cell synapses with a few other stuff. It can synapse with other neurons. It can synapse with another muscle. It can synapse with a gland and activate it. This is what those cells are particularly specialized in doing, is sending signals. So dendrites receive the signal, then this portion will process that signal, and then will send a nervous impulse to the other end here, where it synapses, and then command the next one to do something unique. Some terms. Cell body is going to be the entire portion that you see here, this star-shaped portion here. Cell body. Dendrites are the receiving branches. And up here you can see axon. Axon is this long tail, and this is the only one that sends signals, okay, generally, because it gets a little more complicated. But when you receive a signal from this end, it sends a signal through the axon, so the axon sends things away in an efferent direction. Afferent would be going into it, right? Anywho, so axon. Hillock is going to be this first little triangular space here that pretty much leads to a nervous impulse if it gets triggered enough. The initial segment is where the action potential actually starts. And axolema is simply the plasma membrane of the axon. That's it, just the axolema. The axoplasm is going to be the inside of this space. Everything that's inside, if you were to grab inside and see the juices inside, that's the axoplasm. Axon terminals, these are going to be the locations where one axon meets another object. Again, that could be a neuron, a skeletal muscle, or a gland, or a muscle, period. Muscle, not just skeletal muscles. And so at the very end, these little tail-like extensions are branching from the axon. These are referred as telandria. And these are going to possess axon terminals, and that's going to be the very endings here. And now, so make sure you can identify and label these, and you know what each one of these segments is for. Okay, this is the nice little definitions for them. And what was there to add? And so there's an organization to this, because most cells, right, all of it would just be a cell, like there would be organelles in here, but not really. Here, the cell body possesses all of the organelles for the most part. And everything that's going to travel to this end is going to travel through neural tubules or the cytoskeleton. So it uses the skeleton like an escalator system, and it can actually bring things in and bring things towards away from the cell body. So if we're going away from the cell body, that's going to be the standard way movement. Efferent, away, but then retrograde. Now, when we say retrograde, this means that something up here travels through the axon and goes back into the cell body. Retrograde. And then the last thing to add is, I don't really see it labeled here. Oh, there it is. Yeah. Perikaryon. This is going to be the cytoplasm, which contains all those organelles, perikaryon. And again, that's the space where you find the organelles outside of the nucleus. And do not worry about the cytoskeleton components. These are going to be the same as like intermediate filaments, like neurofilaments. Neurofibrils are going to be the same as neurofilament bundles. But it's okay, you don't need to know these. But just wanted to give you an idea that things just change names. It's the same cytoskeleton components, with a little bit of a difference, but otherwise you just add the word neuro and all of a sudden it becomes something very different, right? And then you'll see here, and this leads me to our next slide as well. Synaptic terminals, that's going to be the endings here. The axon terminals are where they connect. Synaptic terminals are going to be where they synapse. It's almost a play off of the same term. And so with that very thing in mind, here we're going to discuss what is a synapse, what's occurring where it connects. So up here you're seeing your telodendron, right? One of those branches that extend from the axon. You're seeing an axon terminal, and it's synapsing with another cell. And that other cell could be another neuron, another muscle, or a gland. And so that's how they connect. They all resemble just like this. So that's the cool thing, is that you know how the ends of an axon look. And you even sort of already explored it. Neuromuscular junction. You've already learned technically this whole how one end communicates with the bottom end. And so with that, when we say presynaptic, the special word is synapse, connection. Presynaptic means to be before. So you are connected to something else. That else is postsynaptic. You are being connected to. So that's postsynaptic cell. So these axon terminals connected to a postsynaptic cell. So you see the difference? Pre, before, post, after. And what occurs there is neurotransmitter release. You're going to see this again no matter where you go, biology or anatomy, nursing, is that at the very ends of these, you see a bunch of tiny little vesicles that contain chemical messengers. And these chemical messengers, what they do is that they release chemicals into this space and then trigger this cell number 2 to do something else. Triggers the skeletal muscle fiber to contract. Triggers the gland cells to secrete. And so that's sort of what all these ends really look like, which is pretty easy to understand, right? Like that's how they communicate, is that once a nervous impulse, and that nervous impulse is the topic of the very end of this conversation, but we're just really saying once that signal is sent, boom, they communicate. This is the object, right, that location, what's going on at that location. And likewise, remember that we said that this is the receiving end? So when there's a synapse, when there's another cell connecting to this segment, all they do is form this very thing. They form a synapse. And then so they communicate by letting that nervous action potential travel, and then these vesicles, which are called neurotransmitters, will be released and placed into that cell number 2. Okay. And the very easy thing to understand is that if you've understood presynaptic cell, then you understand what presynaptic membrane is. Because that's just the lining or the border of this top one, the presynaptic one. And then postsynaptic is just a border of the one below it. Easy peasy, right? And neurotransmitters are packaged in synaptic vesicles. So all I did was mention they were called vesicles, but you can see that they're called synaptic vesicles because their main job is to submit chemicals to the postsynaptic membrane so that something else can happen to the cell. And then lastly, there are collateral branches. And so sometimes you can see that some branches here, you can see that as we're traveling, here's my mouse. We travel here, we see collateral branch. We will break apart and maybe connect to another cell. Maybe it might even connect to the same one, but for the most part, it probably connects to another cell. This one as well. This one will control some skeletal muscle, but the biceps brachii has a pretty big muscle, so maybe it'll connect it on this part of the muscle, if you were to draw a muscle here. But otherwise it would keep going, connect it. So collateral branches create more axon terminals to make more synapses along some bigger objects. And the only cool information to add here is that neuron replacement, a lot of people think that, if you damage tissue or we don't really rebuild tissue in the brain. That's not necessarily true. In the brain, for your smell receptors, you actually replace those all the time. The retina of the eye for vision, you replace those all the time. And well, not all the time, but sometimes, most of the time. And then hippocampus is there for memory. So when you learn things, as you increase your memory, your joy of learning, you develop cells in your hippocampus. And this has a lot of long-term benefits to prevent, becoming more senile at an earlier age. And also, it's the only place that you really make a connection. It makes you smarter. So learning is really does make your brain go bigger, and everything else kind of doesn't. So that's kind of a cool thing to know, right? And so make sure you know these connections and how to use these terminologies. Here, I'm going to scratch out 2 different axons that I think won't really matter for our sake. I'm a little sad I'm taking them out, but I think it's okay. We're going to take off unipolar. You do not need to worry about them. And you do not need to worry about anaxonic neurons. And after I'm finished explaining these two, you can go back and read it, and then you'll understand the difference, but you will not need to know it for the test. These two are the most common that we encounter in our class. So multipolar neuron, this is going to be the cell body. This is the same one that we've been seeing this whole time. You already know it. It's the one with all the dendrites, one axon, and axon terminals. Now look at this freak of nature here. If you compare it to this one, this is its cell body, and it has a dendritic process that looks like an axon, but it's not an axon. A dendritic process that leads to dendrites. Here below we have our axon, and then we have axon terminals, just like before. So the only difference now is that all the dendrites are not on the cell body, but they're on this little string, a little adapter, which we refer to as dendritic process. And so you can see that it receives a signal, and then it sends a signal through the axon. Same as this one, receives a signal, and then sends it through an axon. And so that's kind of like the same organization, but just a slightly different structure. And the reason is that sometimes there is some cells that their only job is to communicate from point A to point B. These multipolar neurons will have multiple functions, connecting to multiple things, really bringing in, like if you see multipolar, right, you're receiving a signal from this, maybe the eye, then maybe the muscles, then maybe blah, blah, and then this neuron will control the ability to reflex, from something coming right at you, like something flying straight at your face. You're able to control your skeletal muscles because your visual receptors are sent a signal in at the right time. You detected your balance through this. It's just so complicated. But you can do that complicated thing with multipolar neurons. That's really all the take-home is here, is that these are multifaceted. They can receive signals from everywhere and send them out. This just sends signals from point A to point B, probably only connected to one thing, two things, not too many things, but just goes whoop, bloop, bloop, and we'll see an example of that on our final lecture. And so nervous system here, neuroglia, these are the cells that are going to be considered the assistants. Okay, because here I want you to pay attention to this diagram for the most part as I talk. And when you look at this, these blue ones, these are these multipolar neurons that we just saw. And their axons are in here, but now they're wrapped around by a bunch of other neighboring cells. You can see here, you can actually visualize that here. Here's the blue one, the axon. And then you can see all these little leg extensions extending and connecting to it. And we'll talk about what that is. But otherwise, that's the axon that we just saw in the previous slide, the axon. If you're not sure what I'm talking about, step back for a second. Make sure you're aware of your cell terms. Probably shouldn't have been drinking a soda. I'm going to get all burpy, but anywho. Okay, so there we go. Okay, so here, glial cells now. These are going to be supportive cells, and they're going to support those axons and the actual neuron itself, and their axons. And we have a few types of cells to learn. Ependymal cells, as shown up here, and if you look at it onto this image here, you can see that they are still here. This is the same picture that you're seeing up here. And what these ependymal cells do is that they receive blood. Using blood flow in a way, they create cerebrospinal fluid, as spelled right here. Cerebrospinal fluid, CSF. And this is an important fluid in maintaining the health of all your cells, providing the nutrients. And so you have to think about it in this context, that in this you're seeing a bowl of all of this nonsense, and these ependymal cells, they're creating cerebrospinal fluid that's just flowing out in all these spaces. And we'll talk about some other more specifics in a second. And now, next cell, astrocytes. This is probably the better one to jump to. Astrocytes now, what they do is that here we have to pay attention to these. You see these cells here, the ones I'm circling? These are connecting a very important object that you already know what it is, a blood vessel. Capillary. Do we see this? Remember that capillaries are little tiny blood vessels where nutrients and gases that your lungs and your digestive system take in, they bring it into your blood flow, and these astrocytes, they take it in, and then they will pass the nutrients on to the actual neuron cell body and the axons. So they are they form what we know as our blood-brain barrier. They are so good that you can create a miracle drug that helps cure these cells from a disease. But if you put it in your blood vessels like an injection, these astrocytes are so picky about what they allow through that you can't even treat it. Even if it was a miracle cure for the brain, it's almost impossible to get through these astrocytes because of the blood-brain barrier they make. They are little gatekeepers of blood to neurons. And just really interesting stuff. And so, with that in mind, they do a few things. One, they regulate ion, nutrient, and gas concentrations in interstitial fluid around neurons. Now, what does that mean? We understand now that they bring stuff in from blood vessels. What is interstitial fluid then? That's probably the only word that's a little confusing here, right? That means all the fluid that's in this space. All the fluid that you see, like where my mouse is, like if you were to magic school bus in between these, this would all be filled with fluid. That's interstitial fluid. So try to keep in mind that when we talk about ependymal cells, we said that the cerebrospinal fluid flows out and it's pretty much forming like a soup in here. That's part of the interstitial fluid. And yeah. And so they also absorb and recycle neurotransmitters. Where did we see neurotransmitters? At the synapses. Here we're seeing one synapse. Here we're seeing another synapse. Here we're seeing another synapse, right? And those synapses release neurotransmitters onto the postsynaptic membrane or to the surface of the adjacent cell. To the next to this, it had. I didn't say anything wrong. I'm just trying to say it as simple as possible. The one end of this one met the one end of the other one. That end is called the postsynaptic membrane, and that's where the neurotransmitters went. And once they were used up, they go and they flow into the interstitial fluid, and then the astrocytes, as they're flowing through there, they can pick it back up, and maybe even recycle it, or break it apart. It's a little complicated, but otherwise, that's a cool function of the astrocytes. And then they form our scar tissue after injury. And so, with that, oligodendrocytes now. Actually, microglia is a little easier. Microglia, these are going to be monocytes, right? They're very closely related to monocytes. And monocytes, remember that they turned into macrophages. And what did they do? They went around engulfing cells. That's what mobile phagocytic cells means, to be moving around looking for things to engulf and destroy. And you can see that they remove cell debris, like when one of these neurons die, or one of these other little assistant cells die, they go in there and they take it up, they clean it up. They clean up all the waste. These cells, they do produce a waste after they receive their nutrients from the astrocytes that created the blood-brain barrier. Once they used it up, they kind of poop in a way. And so we need these microglia to take care of it. And then they also get rid of any pathogens that make it through that blood-brain barrier, or maybe even some harsh skull fracture injuries. Oligodendrocytes, what they do now. Now, the star of the show here is the axons when you think about oligodendrocytes. The axons of a neuron are the signal senders, right? Signal senders. And so the oligodendrocytes wrap around the axons, and this myelin that it's wrapped around it, this white sheath here that you're seeing, that speeds up the reaction. It also resets it faster so that another one can go through. And so you need this myelin to be around every axon to ensure proper signal conduction, or an action potential conduction. And please remember, you need to start logging away the word action potential, that it means nervous impulse. But action potential is the appropriate word, and we'll see why we have to really break those things apart. Anywho, so this oligodendrocyte wraps around the axons, and they create the myelin sheath, and that's all they do. That's literally their only job. Their job as a cell is to latch on to multiple axons. See, this one's latch on to this one's latch on to this one, a different axon. And then now you have axons with myelin on them. So oligodendrocytes are responsible for that. And again, this myelin sheath speeds up nerve impulse transmission or action potential transmission. And just as a fun fact, before I move on, is that they comprise half the total volume of the nervous system. So that means half of everything isn't just signal sending cells. A lot of them are a lot of supportive cells. So even though I refer to that as the neurons as the star of the show, everyone's got a role to play, right? Just like in life. And so now with myelinated, we're going to examine the myelination now. Whenever something was myelinated, right, that means that it had that protective myelin sheath around the axons. And in the brain, there's going to be a moment when we're going to be talking about white matter versus gray matter. And whenever you're thinking about white matter, that means that you're looking at the bulk of the area possesses nothing but this view. Nothing but a bunch of axons with myelin. So usually white matter in the CNS is always talking about the axons. So if there's issues with your white matter, there's a problem with your axons. And so this space now, what we see when we examine white matter is that we just see a bunch of nodes and internodes. And all that means is gaps. When you look at this myelin sheath, you can see that this is like a one covering, right? It kind of looks like a green bean in a way. And you can see here that this little segment here where it ends and it begins, that beginning of that bracket, there's a little gap right there. There's a little gap right here. And there's another little gap right here if you pay enough attention. If you pay attention to this one's obvious. Here's a gap, here's a gap, here's a gap, here's a gap. Each gap is called a node. A node. A node, 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 node. Internode is each one of these long ones. 1, 2, 3, 4, 5. So each one of these longer sheath, one sheet is an internode. Internode. And traditionally, these nodes were always called nodes of Ranvier. It was always fancy, right? Nodes of Ranvier. And CNS gray matter now. When we say gray matter, it's mainly cell bodies. That's it. And cell bodies typically have dendrites, or they have unmyelinated axons. And they just don't like myelin. They just like myelin because they're not axons. Myelin is for axons, not for the cell body. And so the CNS gray matter, that just means that you're seeing a segment with a bunch of neuron cell bodies. That's it. That's all the difference means between the two. And then the last thing to add for myelination is if you are in the central nervous system, oligodendrocytes were the key star role players in making the myelin. These here, right? These were this one right here. This was our oligodendrocyte. If you're not sure what I'm talking about, look at the last slide. Oligodendrocyte. They myelinate the axons. And so they have a similarity with these other cells because these cells do the same exact thing, the next one we're talking about, and that's the Schwann cells. Schwann cells are also a supportive glial cells, but they're in the PNS. That's the only difference. The last four we talked about in the previous one, ependymal, microglia, oligodendrocyte, and one more I'm not remembering. Astrocyte, did I say that one? I think I said them all. One more time. Astrocyte, oligodendrocyte, microglia, and ependymal cells. Yes, those four are located in your central nervous system, CNS. PNS is all the nerves that you're going to find outside of the spinal cord. Even though they have the same type of nerve cells, the myelin is created way differently. And the only difference is Schwann cells create myelin sheath for the PNS axons. That's it. They do the exact same thing as the oligodendrocytes do. They create a myelin sheath around the axons. The Schwann cells only do it in the PNS. That's the only difference. And do not worry about satellite cells. And now myelinated axons. So now we've formed our myelinated sheath, right? Because all of this, do you notice all this? Each one of these little pegs, 1, 2, 3, 4, 5, 6, 7. That's what makes it so different from the oligodendrocytes, is that Schwann cells are also the myelin sheath. Like they themselves wrap around. Like if we pay attention to this first one here, you can see that there's a little nucleus in here. The other one has a cell, and then it has a little nucleus. So each one of these, one cell creates the myelin sheath just for a little segment of it. And its job is just to be a myelin sheath for the axon. And so the Schwann cells make up the myelin sheath. And the outer surface of those Schwann cells, the surface, like this surface that you're seeing here on the Schwann cell, that's called the neurolema. And so you can see why Schwann cells are actually more appropriately called neurolimocytes. But otherwise, you can call them Schwann cells if you want. Neurolima is the name of the surface. Remember, like sarcolemma, right? That was for the muscles. Neurolima is for the surface of Schwann cells here. And if we move on here, a single Schwann cell wraps around a single internode. So in this case, it's a little bit different now. Back here, we had our internodes were a little bit longer because the oligodendrocytes wrapped around longer regions. But here, only the length of the Schwann cell forms an internode. And I wanted to do PNS, we'll test you on this, but this is just nice reading so you can see now. I think you should all have enough like background to understand what's going on. But I'm going to cut it off from the exam because this next segment is the doozy, and I really would rather you spend more time on the membrane potential stuff. But otherwise, this is a nice little read through it, and if you can understand it, it's a good job. I think you've covered the previous material really well. Now, this is where it gets really hard, so you want to make sure you study really well beforehand and not move on because this part will exhaust you. I don't know. I'm pretty good at explaining it, but we'll see. And I'm just kidding, but anyways. And all right, so in this segment, we're going to take it slow. And so here we have our membrane potential. That's going to be the charge around the plasma membrane of an axon, because that's really the star of the show right now. And the plasma membrane itself of the cell body, even this region here, and also the axon, it undergoes a change. When we talk about a nervous impulse, we are talking about changes in the membrane potential. Now, human speak. It is charged here. Whether you understand charges, positive goes with negative. Some things are very positive, some things are very negative. That there's just a charge distribution like magnets, right? Positive and negative. And so on the outside of the cell of a neuron, it's much more positive. You're always going to see it denoted by a bunch of positives. And on the inside, it's always going to be very negative. So this creates a difference in charge. But this is the way that it always consistently stays. More positive on the outside, more negative on the inside. And we refer to the charges around it as the membrane potential. And now neural activity. Now resting membrane potential or resting potential means a starting point. That if you're looking at an inactive cell, this is what it always looks like. Always has the same charges on the inside and the outside. Is just the starting point. And it's going to be more negative on the inside than on the outside. And the number you want to add here, we're going to see it again, negative 70 millivolts. Keep that number in mind, negative 70. So it is negative 70 millivolts when you check the charge on this. And the reason it's negative is because the inside is much more negative, approximately 70. Okay? And this will matter later. And when we pay attention to our resting membrane potential, that's just what the cell starts as, right? It's undisturbed. But then if it starts receiving a charge or like a neurotransmitters from another cell, right? This can change the resting potential of a cell, and then it creates what we refer to as a graded potential. Now, what you have to think about this is that you have to think about it as a nervous impulse, maybe nervous impulse, because what it is like you don't want your neurons just firing a signal when it receives the slightest little signal. Like it needs to reach a certain level. And that's what this little segment does, is that it doesn't do anything if you give it a little stimulus, but if it doesn't like make a reaction, then nothing happens. But the graded potential, you need to pass a certain limit. And then if the graded potential says, all right, this is enough of a stimulus, then it'll send an impulse, which you know now as an action potential, meaning that this is kind of the gatekeeper. The signal has to be strong enough to make this say, all right, I can send an action potential. And so you can see triggered by sufficiently large graded potential. Now this word potential is going to be what's tricky about this. And whether you were good at physics, it doesn't matter. The word potential means possibility. And I want you to think about it like this. That if you think about can I draw? Yes, I can. If you have a table, okay, and then this is the floor, and up here you have a book. Okay? This book by itself, we say that it has potential, right? Because if I have a book without a table, right, I know that the amount of energy or force that it can provide when it falls, right, I know what that's going to feel like. We all know, we all drop a book, right? You drop a book, you drop anything, it falls at a certain rate. The bigger it is, the more energy it hits the floor with, right? And so that is, we consider, if you hold the book up, if you're just holding it with your hands and not letting it fall, it has potential energy, because you know that if you let it go, it's going to hit the floor with that much energy. So potential energy exists in this book because gravity is pulling it down. And when gravity pulls it down, it's going to hit it with an energy. That energy is called potential energy. That's all that means. So you can see that if you have a table, even though no one's ever, there's nobody in the room, right? And there's just a table and a book. This table and this book still has potential energy, because if the table magically disappeared, it'll hit the floor with a certain amount of energy, potential energy. So when we say the word potential, resting potential, that means that this thing, it has a certain amount of energy it can already go. Okay, something's holding it back. And it always stays at a resting potential, meaning it has energy, but it's not going to do anything. So it's resting. But the second we get to a greater potential, that means that maybe our little table, in our little example here, maybe it's starting to, I'm trying my best to erase here without erasing everything I just drew. Maybe our table now is starting to get a little weaker magically. That's the analogy. It's not an easy one to imagine, but it is. So if we say a graded potential, compare it to a resting, that means that we're getting some stimulus. And so if we're getting some stimulus, that means that this table is starting to break, and this book now is really going to getting closer and closer to really exerting that energy. And now the graded potential, we reach a certain threshold of stimulation, and then bam, all of a sudden, that book is free to fall all the way down to the very bottom and hit the floor. And so that's exactly what you have to imagine. And that's why an action potential, we've reached a certain energy now that's going to provide an action. That means that the action of this falling down, in our case, sending a nervous impulse past this segment to allow it to go through to the next tissues. Next, synaptic activity. That's what occurs when those axon terminals, right, that you learned about, they release neurotransmitters onto the membrane, then they're going to affect the resting potential of the next thing, if it's another cell. And so that's pretty much it. And let's move on. And so now if we take a closer look at this membrane potential, don't let any of this intimidate you. I promise it's easy if you just take it slow. Now, if this is when we measure the resting membrane potential, like we actually put a little apparatus and we check the inside here, you'll notice that when you compare it to the outside, it's negative 70 millivolts. That's what we mean by resting potential. And when you get some movement, like this little segment here, I'm going to label, draw my own little line here, like this. If you measure it there, like if you were actually to see what's going on, like what is an action potential, that all it is, it's the charges changing from the outside and the inside, changing the positivity and the negativity on the inside. And so when you get an action potential, this just becomes much more positive. And we have to think about why that is because if we're measuring the inside now, right, you're measuring the inside of the cell, we know that it's more negative, right? That's a fact that we have to know. It's more negative on the inside, approximately negative 70. That sodium ions, they go in, and when those sodium ions, and this is the illustration for it right about here, well, no, this is not really it, but we'll see it right now in a second. But this, we'll see here a sodium leak channel, that sodium ions can make their way inside. And when they make their way inside, they make this very more positive. So let's reset for one last time. Outside of the axons, outside of the cell body, it is positive. Okay, in the inside, it is negative. If you measure the inside charge, it's negative 70 millivolts. Okay, the outside, it doesn't matter. You need to just keep track of the negative 70 millivolts on the inside. Now, if potassium rushes in, that's going to be these little positive ones. If they rush in, what does positive mean? If you think about negative numbers, if I add more positive values to a negative 70, what's going to happen? The number becomes much more, goes closer to positive. So negative 70, if you go to negative 30, that means you became negative 40 less negative. You're a little more positive, right? This is more positive, negative 30, than negative 70. And then if we go to 0, 0 is much more positive than negative 30. It's much more positive than negative 70. And if we go to plus 30, we can go to plus 70. We can go to plus 40, right? And that plus 30 or plus 40, that's when the signal, that means that things are really moving. Sodium really rushed in to the max, really went in there. And so keep track of that. Sodium movement inside of this area, the cytosol, the inside of the axon and the cell body. When the sodium ions go in, it makes this much more positive. Okay? And vice versa. The inside, if you think about the starting stage of things, before this red line, you notice that inside, up here we have our positive sodiums. That's what the purple ones are. Positive sodium. That's why it's so positive out here. Down here, it's very negative. But the weird and confusing and the only thing that's going to be trippy about this is that potassium is positive. But why is it written as negative in here? And the answer lies in that you have proteins in there that make it very negative. So these proteins, they make it very negative, and so it's negative on the inside. And the positive, this isn't as positive as sodium. Potassium is not as positive as sodium. I know that's a hard fact to wrap our heads around. But like when we were making comparisons up here, we know that this is more positive than this, right? Negative 30 is more positive than negative 70. 0 is more positive than negative 30. Plus 30 is more positive than 0. So in your heads, sodium is more positive than potassium. And so we describe the inside as much more negative, or otherwise negative 70 millivolts by default. The only way that this changes, this whole paradigm, the resting potential changes, the only time it changes is when sodium rushes in, and then this value will go up. This little meter would go up. So now let's wrap our head around some terms before we get into the nitty-gritty of things. We have some tools for the movement of potassium and sodium around this membrane. One of them is going to be our sodium-potassium exchange pump. Do you remember this? What it does is that it lets in it takes out 3 sodium ions, and then it lets in 2 sodium ions. So this is our sodium-potassium exchange pump. And if we pay attention to our arrows, let's start with this one. You can see that sodium can rush into this and turn this whole space into a positive meter, right? It can make this positive meter run if the sodium goes in. But then the cell's like, hey, I need to turn this back into resting potential. So what it does is that those same sodium, it can take out 3 sodium ions, and at the same time, it can take 2 potassium ions and bring it to the inside. So sodium-potassium exchange swap is a tool that an axon uses in order to move sodium to the outside and the potassium to bring it to the inside. So can you see this line here? Maintains stable resting potential. Now, we need to step back one last time before we can really snowball our way into understanding here. That if we maintain stable resting potential, what that means is maintaining this first little illustration here on the side of this line, where the inside is very negative because you have a less positive ion, potassium, and proteins that are negative, always going to be negative 70. So if sodium ions rush into this space, they screw it up. They mess up this whole space. They turn it much more positive because sodium is so powerfully positive. So when it goes inside, it turns this whole space positive. And then you can see that our little needle here, and it's reading the inside. So if it's reading the inside, that means that if sodium rushes in, these purple ones, they rush in, this little needle is going to detect that positivity, and this is going to start moving in this direction, positive. And so the body says, all right, I need these sodium ions out of here. It's too positive in here. So they'll take it out, and then what they do is that they return potassium. So now this is the second part of our story, is that sodium rushes in and potassium likes to come out. And that's what these channels are here, leak channels. You can see that they are allowing potassium to flow into this side. And on this side, the potassiums are flowing through leak channels, which are just flowing in, they're freely allowing them to go in there. But when it becomes too positive, they take out the sodium. Same thing with this one. Potassium, these potassium ions, they like to flow out, and they make this segment way negative, but the sodium-potassium pump is like, nope, I like this side being much more positive, just like we had before. And so what it'll do is that it uses the same sodium-potassium exchange pump, and it makes a deal. It says, take out 3 sodium ions and let in 2 potassium ions. And then it restores everything back to this. Back to a negative 70 resting membrane potential, because this is what it likes to be. And so that describes our next little channel here. Leak channels, they are passive membranes. They always allow potassium to flow out, and they always allow sodium to flow in. They're sort of always open. And this is the reason that you get an interchange between, a lot of this motion is what's creating the charge. But that visualization is a little bit more complicated to get to. And on Tuesday, we'll watch some review videos. So try to absorb as much as I'm saying today. And we'll watch some videos on YouTube that are very useful. And so with that, that's really it. Okay. And then these are always open. Passive transport, meaning that they're always allowing, if it's potassium leak channel, it lets potassium through. If it's a sodium leak channel, it allows sodium to go through. If it's a sodium-potassium exchange pump, it requires energy to force this exchange, force the sodium out and the potassium in to restore back to the resting potential. Okay? And so, maintaining the resting membrane potential, that's really the topic here. And I really wish I would have added it here, but resting membrane potential, that's our topic right now. Is that the star of the show is maintaining that negative 70 millivolts, and it uses the use of sodium-potassium exchange pumps to restore that balance. It uses leak channels because there's always potassium moving in and out to restore and maintain the resting membrane potential. Okay? The mobility of potassium ions to the inside and to the outside is important. Same thing with the sodium. Exact same thing. Now, what about this resting membrane potential? And so this extracellular fluid, the space outside of the cell, has a high concentration of sodium ions, as we've mentioned. Chloride ions as well. Okay, and not so much an important part of our story. But doesn't it make sense? Sodium chloride always like to work together. That's why it's so important to have a healthy salt balance in your body, because salt is an important conductor and maintainer of the membrane potential. And now cytosol. Cytosol is the inside of the cell. This is the inside of the cell body, inside of the axon, and it has a high concentration of potassium ions. We know this already. Potassium, these little potassium ones, and sodium on the outside. And negatively charged proteins, that's these large purple ones. Now, the only reason that this number changes, the only reason you have nerve impulses, like this is the most complicated and the most awesome thing to understand, is that the movement of these two across the membrane of a neuron, that's what a nerve impulse is. There's no electricity. It's just, did sodium go in and did potassium go out? And that's all the nerve impulse is. And let's see that in action right now with our next slide. But let's finish up these facts. And so, this proteins and ions cannot move freely across the cell membrane, that enter and leave only by membrane channels or active transport. That means that these cells, this positive ion, this ion can't just go to the surface and it just can't diffuse through. It always needs to be able to go through its channel. Just like these sodium ones, they can never go through the membrane. They have to go through their channel. That's what this language is by cannot move freely across the cell membrane. That means that they cannot diffuse through. But they definitely can use their membrane channels, which are these membranes, the leak channels, or active transport, and that's going to be our sodium exchange pump. So I would write active transport here. Here, it's already here. Passive transport, passive membrane channels. And so they require, and remember, if anything's an active transport channel, it requires ATP. So, moving down the line. When we measure the membrane potential, we always see that it's a negative 70 millivolts. And the negative implies that it's more negative on the inside than the outside. Look at our little probe. It's measuring it here. Negative. And let's move on. Okay, now, in the grand scheme of things now, that headache of a slide, because I think that's personally your hardest slide, right? That's like, oh crap, like what is it exactly I have to learn from here? Now, in context, this is the inside of the cell. Oh my goodness. And we're back. And so this is the outside, and now the only thing that's left here for you to make everything simpler is sodium and potassium. If you look at resting potential, this is the key star of the show right here. You had positive on the outside and then more negative on the inside. That's what this positive out here means. That's what this negative means out here. Now, when you have an actual nerve impulse, okay, an action potential, a membrane potential change, what's happening is that you are getting, if we pay attention to this graph here, you can see negative 70. That's a resting potential. I keep repeating that for a reason. And if there is enough of a stimulus, like enough of an impulse, like a touch, you touch something that's hot, right? Something cold, you touch it, this will travel down your receptors into the axons, into the cell bodies, and into your brain. And this is how it's sending that electrical impulse or that nervous impulse. It's just letting sodium in from your fingertips, and it's doing so all the way up until your spinal cord, all the way up into your brain. We called that a fact of sending an impulse from resting potential, from nothing to something, as depolarization. And we're going to pay attention to this graph up here. This one's much more easier to understand. We'll go back to the other one. One, a stimulus. A nerve cell wants to send something. It travels up, and then you get a signal, and then the sodium rushes in, depolarization. That's what this note in green is here. Sodium ions flow in. And now at some point, that's all you needed. That's all you needed. Like along the axon, it just travels along a wave. Like if this is our axon, sodium is going in. And then that's enough. Once it sent the signal in one segment, what we have to do is restore the resting potential, because this fact of sending sodium inside of the cell triggered a depolarization or a nervous impulse or an action potential. And then it's traveling, but then now at some point when we get to plus 40, right? We started off at negative 70, now we ended up at plus 40. That at that point you don't need any more. Now it's going to relay it forward to the next part, and then potassium is going to rush in to repolarize. Now, let's examine the word depolarize. Depolarize. You'll notice here we started with a negative charge, right? That's the rules. And the outside we have positive. And then when the action potential occurred, the sodium ions rushed in, and it caused it to be more negative outside, and then it made it much more positive because they brought in their positivity from the outside. That's what we call depolarization. Now, when we say repolarize, that means that we are remaking our membrane back to negative 70. We want to repolarize it, return it back to resting membrane potential. And so you can see that what happens is that potassium leaks out, and then potassium now turns it much more positive on the outside. And then the inside becomes much more negative, the way that they want it to be originally. Right? Like they loved being negative. But that action potential came in and ruined everything. And then it made it more positive. But then the potassium came to the rescue to repolarize. They go outside, turn it more positive on the outside. And then the inside becomes much more negative. And then lastly, now to finish it off, like this is now what you have to visualize. That if the numbers match, this number matches 1. This number matches 2. This number matches 3. And then lastly, this one, number 4, this one matches 4 and 5, match over here. 4 and 5. And so as this travels up the action potential, at 1, you look like this, resting potential. But if the signal is strong enough, and it can lead it to a negative 55 threshold, if it actually sends enough stimulus to make it go there, it'll go up. If it doesn't go up to that threshold, what happens is nothing. Woo, nothing. We're staying at resting state. Too small. Woo, stay at resting state. But then it's enough stimulus, and then now depolarization. That means that sodium comes in, and then potassium rushes out of the inside. It goes out, and then it sends the charges back to normal, even though the concentration of potassium and sodium is different. But that's what this last step is about, 4 and 5. The sodium and the potassium pump, that's what you're seeing here. They lit in 3 sodiums and 2 potassium ions out, 3 sodiums out. They are returning it back to resting potential. And that's what's going on in the graph here, is that when the sodium ions rushed out, that they created a paradigm or a state of existence where now you have potassium out here and sodium. But what's our favorite way to be? What's the resting potential supposed to look like? It's supposed to look like nothing but sodium, nothing but potassium, period. And so we're completely messed up when we're here, right? Depolarization and repolarization. You can see that sodium and potassium are all scrambled. You have sodium in the inside here. You have potassium on the outside here. And we hate that. We want to be back to our resting potential. And so that's where these active transport pump is so useful, because it can very quickly restore it back to resting state by sorting out all those potassium and sodium ions back. It takes all those sodiums out of there, puts them right back to where we want them, puts all the potassium out of there, puts them right back into the cell to where we want them. And then you can see that this is what's occurring, that once we get there, then we're back to resting state, and this would continue forward. And this process, like, it's not perfect, it doesn't go whoop and then all of a sudden stop here. It doesn't stop here, because repolarization is just the fact that sodium ions are moving out, and the sodium potassium pump gets kicked in right about here, and it's so powerful that it brings it right back, or it's able to move all those sodium and potassium ions out to bring it back to resting state. And that's what an action potential is. That's what a nervous impulse is. It's just a domino effect of this. So if you're really struggling, I know that I maybe lost you maybe halfway through here. You'll have to listen through it again. But I would say listen to it a few times so you've understood clearly what I'm saying. And then go watch some YouTube videos on action potential, or just look up resting membrane potential. And I promise a lot of it with the good visuals, it'll be very supportive. I'll try to find some on Sunday and post them for you. And this image, I'm not going to test you on it, but it is important. You've already learned this as well. You're just probably not remembering. But this is like what we examined at a neuromuscular junction. This is a synaptic terminal, right? This is the presynaptic membrane. This is the postsynaptic membrane. And so when an action potential arrives to the end of an axon, it triggers the release of neurotransmitter vesicles, neural vesicles, right? Vesicles that contain neurotransmitters. And then once that action potential, that's what our arrow means here, an action potential finally arrives, and back here it's all those sodium potassium pumps are gonna restore it back to resting once that action potential travels through here. Those sodium ions went in, potassium went out, sodium's gonna fix it all over again. But once it arrives here, it triggers calcium to go in, and then it triggers the vesicles to release neurotransmitters at the presynaptic membrane. And then those neurotransmitters will arrive to a postsynaptic membrane. They will bind to a receptor and trigger a result in something else. And in all likelihood, it's just sodium. And now don't you see the trend that sodium was that first signal telling something, hey, I'm the beginning of something. Because in the beginning, nothing was going on, but then sodium goes in and bam, something begins. So normally when we're talking about nervous impulses, nervous communication, it usually means that sodium is flowing into something, like in our action potentials previously. Make sure you understand the previous slide on membrane potential, what the chart means, 1, 2, 3, 4, and 5. It'll be the subject of our review, and make sure you study all the terms beforehand that are in bold. And we'll review all this ending material on to start Tuesday. Study this hard, move on to the next segment, the second packet.